Biology students, welcome back. We are starting a new unit today on cell division or cell reproduction. We're going to cover a few main topics. Today we're going to look at mitosis. We're then going to move into cancer and stem cells. Then we walk through the process of meiosis, and then we end with karyotypes and chromosomal disorders. That wraps up this unit. So today we're going to look at mitosis, or like I said, cell division. It's there's quite a bit of information in today. I'm going to try and cover it all and keep this video fairly short in the process. So if I move too fast, please, I will probably move too fast for you to write things down in your outline. Please pause as you need to, go back, rewatch to make sure you're gaining all the information that you need. So mitosis, like I said earlier, is cell division. So it's dealing with our cells. Remember, all living things are made of one or more cells. We are made of between 50 and 75 trillion cells. The average human falls anywhere between 50 and 75 trillion cells. We produce 100 to 200 billion cells per day, whether that's new hair cells, skin cells, blood cells, you name it. We're constantly replacing old, worn out, dying or dead cells and replacing them with new cells constantly. So this process of mitosis is simply that. It's making new cells from these old, dying, worn out cells, which your body is constantly doing. Most of your cells reproduce in this way, in this mitosis way. The only cells in your body that do not reproduce in this way are your sex cells, your sperm and egg cells. Every other cell in your body is going to replicate using this process of mitosis. And it's literally just a copy machine. You're just taking one cell and it's running through a process to make an identical copy of itself. Some other numbers up there, again, just the rate at which some cells are replaced. You've got your hair cells, how much are replaced daily, which amounts to about 6 inches a year. You've got skin cells that are constantly being replaced. Like I said, blood cells, stomach cells, you name it. Question here on the bottom, how many chromosomes are in a human body cell? Most of your cells, again, aside from your sex cells, are consisting of 46. Other organisms, other animals vary in their chromosome number, but human cells have 46 chromosomes. Your sex cells, your sperm and egg, only have 23, but every other cell in your body has 46 chromosomes. So why? Why do our cells need to undergo this process of mitosis and division? The three main reasons are as follows. Reproduction, so those are your sex cells, which are going to be in the process of meiosis, which is coming up in a couple days. Repair and growth. So the repair and growth are the two we're going to focus on in mitosis, because you're just literally repairing and growing those cells by replacing the cells you already have. So just making identical copies to repair damaged tissue, damaged cells, or just growth of the individual. Why are cells so small? So we know cells are small. Obviously, they have to be if we're making, or replacing millions of them every day. We're made of trillions of them. What is the deal with them being so small? Their small size has a reason. The reason cells are small, or the purpose of them being small, is because that small size increases their efficiency. If you look at this image down here, if you have a small surface area, so surface area is the area that covers the outside. If you have a small surface area, that allows material to enter and leave the cell more quickly at a quicker rate because there's less space in the cell to move around and locate wherever they're going. Versus if you have something entering the cell in a larger cell, it needs to travel farther to get to that nucleus or to get to whatever it needs to do inside that cell, and then also longer to leave just because there's more space. So cells need to be small to increase their efficiency so they can work more quickly and like I said, be more efficient. Another image is up here, same idea. The smaller the surface area of the cell, quicker and more easily material can enter and leave the cell and get its job complete. I use the analogy in class. typically of a fire, right? If you have a small little house and there's a fire inside, you're able to exit that house more quickly because there's less surface area, there's less inside this house. Versus if you have this very large castle, mansion thing, whatever this is, and there's a fire, you may not be able to exit as easily. That's my analogy. DNA. So if we're talking about cell reproduction, we need to talk about DNA because DNA remembers the material inside the nucleus. The DNA is actually what's being copied, split, and made into or put into new cells. There's 3 forms of that DNA. They're termed chromatin, chromatids, and chromosomes. It's all DNA. It's all the same DNA, but the look of it and what it's doing is going to change. Therefore, the names of what that DNA is called is also going to change. So chromatin, I'm going to use this image down here. Chromatin is a stringy spaghetti-like material inside the nucleus of a cell. Most of the time when the cell is just kind of hanging out, doing its normal job, its normal function, it exists in that form. Chromatin exists in just that loose, relaxed form. As the cell moves to division or to be replicated, this DNA needs to wind up tight so it doesn't lose pieces of it. So it winds up tight into chromatids, which kind of look like this X down here. Each side of this X is an individual chromatid. One side is an exact copy of the other side. So we call these sister chromatids because they're identical. Chromosome is that whole entire piece, that X-looking structure. The whole structure is called a chromosome. If you look over here, here's your stringy DNA, chromatin, relaxed, hanging out. Each individual strand or side of that X is called a chromatid. Sister chromatids are identical, so this side of the chromosome has identical DNA to this side. This entire structure, this entire X is called a chromosome. Held together, those sister chromatids are held together by a centromere, this little circle structure in the middle. So a lot of C words to try and keep straight here, but all related to DNA, all the same DNA, just depending on what part of the cell cycle it is in is going to determine what term we use to describe it. All right, moving into the actual cell cycle. All right, so what's the life cycle of a cell? So as it's moving through your body and doing what it needs to do and then replicating or duplicating itself, what does that look like? So there are 3 main steps in the middle of this diagram. We have interphase down here, mitosis, and cytokinesis. Interphase makes up most of the cell's life. Cell cycle life is interphase. Interphase is made up of, or broken up into 3 steps, G1, S, and G2. G1 is the longest part of interphase. Okay, G1 is just, or gap 1 it's called, is just the cell doing its normal job, its normal function. A skin cell is just doing skin cell jobs, a blood cell is doing a blood cell job. Once it begins to age, it then moves into S, the S phase, or synthesis. S stands for synthesis, where the DNA is doubled. So right now, if we're talking about a human cell, we have 46 chromosomes in the cell. It's just doing its thing. As it moves through S, it now doubles to 92 chromosomes. It doubles in DNA because it needs to prepare for that division. And it wants to keep its own DNA and give a copy of the DNA to the new cell. Once that DNA is copied, it moves through G2, which is just a resting stage for the cell before it enters mitosis. Mitosis is when the cell is actually going to divide or go through the process of division. And then this last piece, cytokinesis, which is the shortest piece here, takes the shortest amount of time, is literally just the cell physically dividing into 2 separate cells. So we'll walk through these steps. Again, take time to look over this, look over these terms, know the order of them, what happens, you know, before, what happens in each step, and what happens in the order that it does. So G1 comes before S, G2 comes after S, etc. So know the cycle and the steps in order pretty well. We're going to move to talk about the stages, the specific stages of mitosis up here. So here are your stages: prophase, metaphase, anaphase, telophase. And then your last step, which I kind of put down here because it's not really part of mitosis, it's its own step in itself, is cytokinesis, the actual dividing into two separate cells. So we'll walk through what's happening in each one of these steps, but this is an overall image of what mitosis looks like in your cells. You'll notice in your outline, if you're following in the outline, I have the descriptions of what's happening in each of these steps already listed for you. You need to either draw a picture or add an image of the stage into your outline. So prophase is the first step of mitosis. And some people use the acronym. Let me go back here a minute. PMAT, P-M-A-T, is one way to remember the order and stages of mitosis. So PMAT. So we're going to start with prophase. The nuclear membrane. Okay, so remember the nuclear membrane surrounds the DNA. That's represented as these white little dashes right here. The nuclear membrane has to go away because the DNA needs to move. And it can't move if it's trapped inside the nucleus. So in this stage, the nuclear membrane disintegrates. The chromosomes condense, so they wind up tight into those chromatids, right? So here you've got a chromatid connected to another chromatid by a centromere, making a full chromosome. Spindle fibers begin to form. That's what these yellow lines represent. So spindle fibers shoot out from these. They're called centrioles. On each side, they will be used in the next two stages. So prophase, nuclear membrane disintegrates, and chromosomes condense. Metaphase. Metaphase, they line up. So those chromosomes line up in the middle. Metaphase, middle. They both start with an M. This is the most easily recognizable step, in my opinion. It looks like just a line going down the middle of chromosomes. So they line up along the equator is what we call it, or along the middle of the cell. Again, these spindle fibers are attached to the centromeres of each of the chromosomes. That will be important for our next step. After metaphase, we've gone prophase, metaphase, we now move into anaphase. Anaphase is when the chromosomes are pulled apart. Anaphase, apart. We'll start with an A. Anaphase, apart. So metaphase, they've lined up in the middle. Now these identical chromosomes, the identical DNA is being pulled apart. Half to one side of the cell, or one pole of the cell, the other half of the chromosome to the other pole of the cell. So anaphase, apart. Excuse me. Telophase. The chromosomes have arrived at their poles. So the chromosomes are where they need to be in each of their cells. The nucleolus, or in the nuclear membrane, starts to reform. So there's those white dashes. The nuclear membrane is starting to come back to form around the DNA. And now instead of the DNA in chromosome form, the DNA is relaxing back into chromatin. Remember, that's the relaxed state. And you'll start to see the cell beginning to pinch in the middle and start to divide. It hasn't completed division yet. That will happen in cytokinesis, but it's beginning to show the progress towards actual cell division. So the chromatin is now home. It's where it needs to be, and it relaxes back out into chromatin form. Our last step is cytokinesis. So here's cytokinesis. You have the actual complete division of the cell into two separate identical cells. You're just making a copy of the original. In animal cells, they pinch in the middle. Okay, so they're kind of soft and squishy. So this middle literally just pinches in half and you have two identical cells. In plants, because of that cell wall, it is not soft and squishy and you're not able to just squish it. So instead, a cell plate forms in the middle to divide those two cells. So those are the steps of mitosis. The cell cycle above in that image, know those steps fairly well of interphase, the G1S, G2, and know the different steps of mitosis. Overall, going from one cell to just making an identical copy of that cell into more cells. Your body constantly does this to maintain the cells in your body. As always, feel free to ask your teachers if you have any questions.